Hello everyone, this is Dr. Santosh. So please subscribe this channel, like this video, press the bell button. Today we will be talking about the topic uh, called RNA editing and that significance of RNA editing for formation of this ApoB48 protein in intestine, and that this ApoB48 protein is very essential part of chylomicron. It is integral protein of chylomicron, and that is being produced in intestine after we eat fatty food or food. And then let's see what happens in our body. We have after human genome project, our body has we found that about 20,000 genes are there. But if we compare number of gene with the protein, so protein number of protein is much higher than the number of gene. And we have one hypothesis and one theory, and that is called one gene one protein. So if we see that theory and number of protein, so this theory is not followed. But it is not like that, because proteins only mRNA that is being produced from this 25 gene is about 25 mRNA, especially heterogeneous mRNA will be produced. What happens is this heterogeneous mRNA will go under the processing to form mRNA, mature mRNA, that is ready for translation. And then after translation also there will be various processes. So there is post transcriptional processes that converts this heterogeneous mRNA through splicing into mRNA, and mRNA will also go under certain processing, capping of mRNA and tail addition in mRNA in our body eukaryotic cells. And those mRNA again will form protein, and protein will again undergo post translational modification. And this all modification, because of permutation combination, they will generate huge number of protein. It has capability to generate huge number of protein. So that is why this post translational modification is also an example of isoform proteins are present in different tissues. Now we will now focus on one of the mechanism of post transcriptional modification. So post transcriptional modification, we will talk about editing of mRNA. This is very good example and very good concept to understand this post transcriptional modification. One of the post transcriptional modification is splicing. Through splicing also many proteins are generated, but here we will talk about editing of mRNA. Editing of mRNA, that because of that editing of mRNA, same gene produces 48 B48 ApoB48 protein, and in liver there is no editing of mRNA, and that produces whole big chunk of protein B100. Now let's talk about how does this editing happens. So this editing happens in intestine. In liver there is no editing. So in intestine how does this editing happens? So editing happens at the at the genetic codon in between this long mRNA. So at genetic codon it will happen. So in middle of the genetic codon, so there is one codon. That codon is called CAA in normal condition. So this CAA is actually this is codon for amino acid glutamine. So what happens in intestine is this C will convert into UAA. So what is this U? How does it convert into U? So it happens through one enzyme. It is called cytosine deaminase. You can see the difference between the structure of this. This is uracil, this is cytosine. The structural difference between this uracil and cytosine is amine group. So when amine is removed, so oxo group will be added, and this will form uracil, and uracil UAA is a stop codon. Now when translation occurs in intestine, so what will happen is the translation will stop around here only. Translation will stop around in between only, and because of this there will be half protein is only translated, and this form B48, like 48% of B gene. This is 48% of B gene. So this is B48, and this is very important part of chylomicron. And let me tell you, this B100, this has about 4000 amino acid. Now this will have about 2000 amino acid. So this is how B48 will be generated, and it does not have carboxy terminal like this. And one very significance of this B48, because this does not have this carboxy terminal, this B48 cannot act as ligand of receptor in liver. So B48 is function only as um, part of chylomicron, as integral part of chylomicron. It only perform function of carrying lipid. It does not help in receptor docking. But here in VLDL, B100 is produced, and this B100 will carry lipid also, and this B100 carboxyl has enough capacity. It has amino acid arrangement that will interact with, ultimately with BLDL, will convert in blood into LDL, and LDL will interact with LDL receptor, this carboxy terminal. So this LDL can uptaken by liver through this carboxy terminal, which is not there in chylomicron. So therefore chylomicron will require extra protein E, for receptor docking. So this is all about this difference between, this is very important concept, RNA editing. This RNA editing will produce ApoB48 in intestine, which is very important part of chylomicron. This is very important concept, you need to understand. That is why I have made separate video on. Thank you for watching. Please don't forget to subscribe, like this video, press the bell button. If you have any question, please put in the comment box. Thank you.